By following the procedures that I have given in this protocol, you will be able to perform plating procedures without contaminating media. Then isolate single bacterial colonies by the streak plating method. And also I have included pore plating and spread plating methods to determine the concentration of the bacteria. So all of these procedures are being included in this video. So let's get started. Now, before starting the procedures, one thing is very important, that is labeling. So label your plates. Label around the edge of the bottom and not the lid of an agar plate with at least your name, the date when you are performing the protocol, the type of growth medium in which your microorganism is growing, and the type of organism to be plated on the medium. Another important thing is that the plates must be completely dry without condensation on the lid. Streak plate procedure: isolation of bacterial colonies using the quadrant method. Now the streak plate procedure, why do we use it? We use it to isolate the pure cultures of bacteria from the mixed population, where you have different cultures of bacteria, different types of bacteria. So when you need to isolate the pure cultures, you use streak plate procedure. Now coming up to the procedure, for the first quadrant, what we are going to do is that we are going to pick up the sample either from the suspension, from the broth, or directly from the colony present in the agar plate, and then spread over about 1/4 of the surface of the medium using a rapid, smooth back and forth motion. Now this back and forth motion is something which is of great importance during this technique. Then for the second step, what we are going to do is that after we completed the first quadrant, now we are going to invert and set down the plate back into the lid on the bench, and then again reflame the metal loop. Now turn the petri plate at 90 degree to streak the second quadrant. Lift the bottom half of an inverted plate from the bench and then touch the loop to the first quadrant near the end of the last streak. So what you are going to do? You are just going to touch the last streak in the first quadrant and then start with that, right? Again, you are going to use the back and forth pattern. Cross over the last half of the streaks in the first quadrant, then move into the empty second quadrant. Again, when you are finished with this quadrant, invert and set down the plate back into the lid on the bench. Now for the third and fourth quadrant, you have to repeat this third step again and again. With the back and forth motion, you have to again get the new streaks. As soon as you have finished with this quadrant streaking, what you are going to do is that you are going to incubate these plates upside down. Now the question is that why is the need to incubate the plates upside down? The point is that we do not need any condensation to be accumulated on the lid so that it does not drip on the colonies, right? So now talking about the advantages and disadvantages of this procedure. Advantages: it includes that it is useful when you need to separate organisms in a mixed culture or when you need to study the colony morphology of an organism. Right, so this is about the advantage, and when we talk about the disadvantage, you will not be able to grow any obligate anaerobes using this method because there is growth of microorganism on the surface, so definitely only those which are aerobes will be able to grow, and obligate anaerobes which do not which cannot tolerate oxygen, they cannot be grown, right? And secondly, only organisms that are viable will be able to grow by this method. So these are about the advantages and disadvantages of the streak plate method.